On behalf of my co-presenter, Dr. Luis Camacho, and myself, we would like to welcome you to the program Pinpointing the Top Questions and Considerations about Biosimilars in Hematology and Oncology. I am Dr. Sanjeev Agarwala from Temple University in Philadelphia, Pennsylvania, Professor of Medicine and Chief of Oncology at St. Luke's Cancer Center. Dr. Camacho?
Hi, Sanjeev. It's very nice to join you for this educational activity. I'm Luis Camacho. I'm the current director of the Center for Oncology and Blood Disorders in Houston, Texas.
First, I'd like to go over the learning objectives for today. Our first objective is to define what a biosimilar is, talk about extrapolation of indications and interchangeability on the basis of FDA definitions and guidance. The second objective is to explain the similarities and differences between biosimilars and the reference originator biologic, as well as the difference between the term biosimilar and generic. The third objective is to evaluate key aspects of current FDA guidelines that inform the healthcare provider's knowledge of the biosimilar approval process, manufacturing, and naming, as well as state regulations surrounding substitution practices. And the fourth objective is to apply foundational knowledge on biosimilars to clinical situations relevant to hematology-oncology practice. As I think we're all aware, the list of approved biosimilars in the United States is growing quite dramatically, and shown on this first slide is what I would say a partial list of the biosimilars that are approved, because this is changing all the time. But as of today, we have these biosimilars available, or at least approved for us, and they include drugs like Filgrastim, Sandoz, Zarcio, which is similar to Neupogen, Infliximab, similar to Remicade, Etanercept, similar to Enbrel, Adalimumab, Atto, which is similar to Humira, Infliximab, Abda, Remicade, Adalimumab ADBM, which is Humira again, and then most recently Bevacizumab AWWB, which is Avastin. So I'm sure you're all familiar with the reference products, and as you can see, we'll talk about naming, but the names of the biosimilars are listed here along with their years of FDA approval, and expect this list to grow quite quickly. So a quick review of the core concepts. The first most important point is that a biologic, including a biosimilar, cannot really be made generic, and we'll explain this more as we move forward. And the reason for this is that these are complex molecules. There's complexity, and I think most importantly, there's variation over time, which is natural. It is impossible for a biologic to be exactly the same from batch to batch and month to month and year to year. So this is a natural situation with biologics, and this includes biosimilars. Secondly, the definition of a biosimilar is that it is a biological product that is highly similar, not identical, highly similar to a U.S. licensed reference biological product for which there are no clinically meaningful differences in safety, purity, or potency of the product. Every word of this definition is important, and the highlighted words are even more important, and that's really important to understand in terms of what we're going to talk about as what biosimilars are and how they are used in oncology. And the third core concept is that the comparability exercise used to demonstrate that a biosimilar is highly similar to a reference biologic is very well established. It is scientific, it is robust, and it is regulated. So this is not just an arbitrary process, but it is a very well-defined process to demonstrate this similarity. So one question that keeps coming up in everyone's mind is, how concerned should clinicians be about the quality of a biosimilar versus the originator or reference biologic? Louis, I'm sure you've faced this repeatedly. What are your thoughts on this, and how do you address this when you talk to people?
Sanjay, I think this, including myself, has been a great educational process. From the time we started talking about biosimilars, I came to realize how many batches of biologicals we go through and how much these molecules that we give to our patients on a daily basis can change since their initial approval. And I think that's something very important for all of us to know. Like you mentioned before, we need to understand that these are not generics. So one cannot expect the chemical structure to be identical over time. These are molecules that are developed by living organisms and as such, they have variability between lots, between batches, and therefore, I think that once the biosimilars are endorsed by the regulatory agencies, we can expect certain variations from themselves and from the originators.
That's a very good point, Louis, and that's exactly what we're going to try to show in this program is that there really should not be a significant concern because there's really no major difference, clinically meaningful differences for the definition between the biosimilar and the originator product. So thank you for that. The other thing that I think is very interesting to realize, and most people don't know this, and as Dr. Camacho said, this has really been an excellent learning process, even for us who deal with this every day, is that there are many post-approval life cycle changes to the reference biologic. And this is kind of what Dr. Camacho was talking about, that over time, there are changes that take place naturally. And some of these changes can be quite significant. And actually, some of these changes are even more than what they are between the reference biologic and today's biosimilars. So, for example, here, infliximab, just the reference product infliximab, has gone through more than 35 changes in the manufacturing process after approval, yet it's the same drug and has the same efficacy. And this should give us reassurance regarding the biosimilar process, approval process, and how these drugs work. And here's another example of trastuzumab, the manufacturing process, the changes, the drifts in the quality attributes. And I'm not going to go into detail here, but you can see the slide. There is difference in the biological structure that changes the activity and the antibody binding, and none of this is clinically significant, and this is just a natural process, drifts, in terms of this one drug, trastuzumab. So the question really comes up then is, what is acceptable variation? How much is acceptable? In other words, you cannot have a process where every biological agent from batch to batch has to be reapproved. So this has been established as to what the lot-to-lot variation of, and here an example of Rituximab, of the Innovator product is over time. And as you can see from this chart, there are changes again in the chemistry, the structure, and of course in some of the biology, and even the ADCC, but none of this matters in terms of the efficacy of this product for the patient receiving the drug. We mentioned this before, but I'll repeat it. Identical copies of a biologic just cannot be made. It is impossible, just given the nature of these biological molecules. As Dr. Camacho mentioned, they are made by living organisms. It is impossible to make identical copies, and that's the difference between really a biologic, a biosimilar, and a generic, and we'll talk about that. Reference biologics change naturally over time. There is variation over time. The manufacturing process changes over time. These are normal, and they are highly similar to each other. In other words, the different batches of the reference biologic are highly similar to each other. Therefore, if you can manufacture a biological agent that is also highly similar, that can be a biosimilar because that is highly similar to the reference biologic and therefore has about the same variability as the original product would have over time. So here's the differences seen quite nicely in terms of generics versus biosimilars. So generics are exact copies. They're like carbon copies, Xerox copies as we call them. Chemical structure, you make it the same, it is the same. The approval process of the generics is done by the FDCA, the Food and Drug Cosmetics Act, that basically identifies a process where and how a generic can be approved. And this is outlined on this slide. There have to be identical to an approved product, and there really is therefore no safety and efficacy data required because they're identical molecules. A biosimilar is different. It is approved by a different process, by the PHSA, which is a Public Health Service Act. And the point here is, as we've discussed, they are not going to be identical. They're going to be highly similar. So the point is they need to have data showing that there is no clinically meaningful difference between this biosimilar and the reference biologic in order for approval to happen. And you can see the process outlined here. So here's another question, Louis, in terms of, this whole process. And we as clinicians, you and I go back a long time, and we know and we're very comfortable with generics and the process of approval. When a generic is approved by the FDA, we don't think twice about it, But the biosimilar approval process is a different process, and I've just gone through it. But in terms of reassuring our colleagues, is this process rigorous enough? And what do we know about the FDA approach to the approval of biosimilars?
Sanjay, I believe, and again, this is my experience, that it's a process that is mainly led by trust, okay? I think we are extremely lucky and blessed to have an agency like the FDA regulating this process because we trust in the regulatory pathways. I think the EMA is also doing an excellent job, and I think that time will spread those requirements around the globe and come to, international guidelines for their approval. I believe the process as it is a constant tune-up, but I think we have a very good startup. You will talk later on as to how many products have been approved in Europe. But I also think that part of it is the sponsor developing the biosimilars. Part of it has to do with our education as physicians and healthcare providers, the experience that we gain with them, and, of course, the pharmacovigilance. So I think that all comes to close as a process. And I believe nowadays it's rigorous enough to my taste. What do you think?
Oh, I agree, Louis, completely. In fact, I would also say that the FDA sometimes has been accused of being too conservative. And I think, as we all know, we are very much behind the curveball. You mentioned Europe. They have approved so many more than the FDA has. So I think I agree with you 100%. Our process through the FDA, we are blessed to have an organization that is actually very rigorous and very compulsive about the process to making sure that patients are protected. So I'm going to go through here a stepwise approach, to the evaluation of biosimilarity as a concept for the approval process. So one thing to realize, it's a totality of the evidence. It's all this evidence put together that leads to the process of approval that, as Dr. Camacho and I have talked about, the comfort level to make sure that we feel that these biosimilars are not clinically meaningfully different from the reference product. So the first step, and you can see the left side of the slide, analytical studies are done to look at the structure. The animal studies are done after that. Then clinical PK and PD studies, very important. Immunogenicity, biological agents have this unique ability to produce antibodies, and that's a very important part of the process to make sure that it's covered. And then additional clinical studies as needed. And this all evidence together is the type of biosimilar development program to demonstrate the biosimilarity, to make sure that there is no clinically meaningful difference between the biosimilar and the reference biologic. Here you can see, I think, explained nicely in this sort of pyramid triangle shape depiction of how this process works. So you start with the foundation of the analytical foundation, which is the most important part, obviously, because you got to create the molecule that's similar. Not identical, can't be, but very similar. Then you do the non-clinical studies, which are a little less important. The clinical pharmacology comes next. And as you can see, the additional clinical studies, although important, are the smallest piece of this triangle. And that's very important because we'll talk about this, but there need to be a certain amount of studies. But the whole point of the biosimilar process is to allow drugs to be manufactured that are similar, biosimilar, lower the cost, and therefore we do not need to do every single study all over again. If we did, we would be defeating the entire purpose, and the cost of developing these biosimilars would be the same or even more than the reference product, which would be completely counterintuitive. So there is a whole balance here of what data is needed to prove that the biosimilar is not different, no clinically meaningful differences for the approval process. So here's a slide showing you some of the comparative analytical data. You've got the structure analysis, the primary sequence, the amino acid sequence. As we know, these molecules are very complex, so the higher order structure, including aggregation. There is post-translation modifications, glycosylation, phosphorylation. These are biochemical processes and other variations, modifications. That's analyzed very carefully. And then there is structure, of course, but there has to be function. So the function is the in vitro assays, including biological binding, enzyme kinetics, and then in vivo studies in terms of the animal models of disease. So this is the whole basis of the analytical data that is done in this process. So here's an example of a biosimilar and Innovator Filgrastim drug. This is U.S. and EU, and you can see here the protein characterization, receptor binding studies depicted here. And as I mentioned, the analytical structures are really the foundation of the whole process. So this is the bottom of that pyramid, the bottom of that triangle, the largest chunk. And if extensive structural and functional comparability assessment does not reveal any significant difference between the biosimilar and the reference product, it is highly unlikely that the clinical trials would show any difference. Still, some clinical trials are done, just to be sure, but it would be very unlikely there to be a difference. That's logical. So clinical studies of biosimilars, how are these done? And we all know, most of us are involved in clinical trials, so we know how complicated they are. So the idea is here, the major difference is when you're doing a biosimilar trial, you are really trying to show that the proposed biosimilar, the proposed product, is neither better nor worse, neither inferior nor superior. This is not a trial to show that it's better. And those are the trials that, Dr. Camacho and I, for example, do all the time. New drug, trying to show it's better than what we have to improve cancer care. This is different. The concept here is to make sure that it fits that specified margin, neither inferior nor superior, the balancing act. And therefore, the endpoints and study populations selected are those that would be clinically relevant and sensitive in detecting these clinically meaningful differences should they exist. So those are the kind of studies that we do in this situation. Going back to the example of Filgrastim, biosimilar Filgrastim and reference Filgrastim, you can see these two lines basically completely overlap, and you don't need a statistician to tell you that there is really no difference here in the PD analysis or the PK analysis between the reference biologic and the biosimilar Filgrastim. In studies of severe neutropenia, duration of severe neutropenia, there have been clinical trials done and have shown that the biosimilar Filgrastim is non-inferior to the reference product in terms of the duration of severe neutropenia. And you can see the study design specifically. This is relating to Zarcio, the Filgrastim from Sandoz. Here's some more data to show you. Again, you can see the curves basically overlap. This is the time course of the absolute neutrophil count in cycle 1 chemotherapy for breast cancer, myelosuppressive chemotherapy, where this kind of treatment with Filgrastim will be standard. And you can see that the biosimilar product and the reference product really are overlapping, and there is non-inferiority of the biosimilar in terms of the reference product. This is cycle 1 absolute neutrophil count. And you can see, again, the details in more than 200 patients. So, Louis, back to you in terms of another question. Are there any specific safety concerns with biosimilars? And we're talking about Filgrastim here, but can you give us your perspective on any kind of safety concerns with biosimilars?
This clinical trial was a beautiful experience of a study that randomized in 4 different arms. These were for patients with breast cancer, locally advanced, who underwent neoadjuvant chemotherapy, and they were randomized to 3 cycles were given with the reference product. The first arm was all received the reference product. Second arm, all received the biosimilar. And then arms 3 and 4 were alternating. And it did not show any differences. I think that I do have one concern. The one concern that people have expressed repeatedly is that these clinical trials may be too small to find signals of safety that would set off the alarms. However, what you were mentioning earlier was that in that pyramid diagram, where we have more than 50% at the base looking at the analytical, which is the foundation data, I think that if you have those that prove very, very close to the reference molecule, chances are that you will not find them. I mean, we still leave something out of the box, and we will both refer to that point later, and that's the issue of immunogenicity. For the most part, I think that one could be reassured that it's a safe process. It's a safe conclusion at the end of the day when you find that there are no major signs of differences between both agents.
Very good point, Louis. And, going back to what we were talking about earlier in terms of just the reference product have the same variability, exactly right. So I agree. I don't see any major safety concerns. And, again, the pyramid structure we talked about is crucial because that is the entire foundation of the process, which gives us reassurance that if it is so similar in terms of structure and therefore function, you would expect there to be this similar efficacy and safety. Here's actually some data on the biosimilar Filgrastim safety to that point. This is a phase 3 trial, and Dr. Camacho already described this trial to you. I thought this was very important that there was no patient who developed a neutralizing antibody in any treatment arm, which is important because one of the concerns, as Dr. Camacho brought up, and he will talk about later as well, is the immunogenicity issue. That's a big concern because if you have some variability in the structure, even though it's similar, could there be difference in immunogenicity, which could be clinically meaningful or not, depending upon the product and the disease? And then the treatment emergent AEs were basically identical. As you can see, 20.6 versus 19.6, no difference. So to Dr. Camacho's point, there was significant data here that this was very safe. Here's another study, a monitor GCS study, which is an international prospective open-label observational study, and it's a large number of patients, so it gives us some more data in terms of a larger group of patients, more than 1,400, almost 1,500 patients, and these are patients treated with myelosuppressive chemotherapy, and once again, the point here to make is that there really was very similar consistency with the safety profile for GCSF in this particular trial, not showing any clinically meaningful differences in terms of safety to drive that point home.
So, Sanjay, the question that I have for you is, this issue about extrapolation, and it essentially refers to whether or not the biosimilar is approved for the same indications and labels that the reference already has established. How do you feel about this? And before we enter that subject, do you have any concerns in that regard?
That's an excellent question, Louis, and I know you're going to cover this in detail. I think, to me, this may be one of the most important issues that at least I used to face, and I'm much more reassured now, and then my colleagues are going to talk about it, because this whole concept of extrapolation sounds, very scary. But I'm no longer concerned about it at all, because extrapolation is a logical process, a thoughtful process where when we have gone through the system that we've just talked about in terms of the approval process with all the structural similarities, the data, et cetera, and then you do a clinical trial, a limited, smaller trial just to show non-inferiority in a particular disease area, I don't think there's really much concern in extrapolating that to other indications that the original reference product has. In other words, the biosimilar should be, logically, if it works in one of the diseases where the reference product is shown to be effective and approved, why would not it work in the others? Now, there may be a few exceptions to that, and I would love your thoughts on this, but I really don't think so. I think as a logical human being, I highly doubt there would be a difference.
Yeah, I'm on the same page with you, Sanjay. I think this one explains what the concept of extrapolation is. And this was essentially developed by the FDA in 2016. And it's something that, as plain and simple as it sounds, and here's sort of how the framework reveals in the process. This is taken from the EMA website, and you see how on the leftmost part of the slide, there's the totality of the evidence, and you see again the triangle that Dr. Agarwal presented to us earlier. And based on those data, those are the data that are submitted to the FDA, they move to the reference biologic experience. And they compare, and they see if they feel that the totality of the evidence is in support of other patient populations. For example, if you are developing Rituximab for low-grade lymphoma and you want to get the indication for rheumatoid arthritis, do we have plenty of evidence that make it biosimilar? Remember, at the end of the day, this is about similarity. And then you move on to the scientific justification, and you see the different populations. And it's best if objective eyes, unbiased eyes, look at the data and tell you if you have plenty of information there to apply or get extrapolation. So I think this is a concept that is very important to get, that until very recently had plenty of concerns and controversies. And here's part of what we were talking about. Essentially, you see that once you have very strong non-clinical, analytical PK/PD data, then you can go to other patient populations. You see the features of these studies are, usually having very strong scientific data with very limited number of patients. And as Dr. Agarwal was mentioning, you cannot make them larger in an attempt to find signals of unsafe behavior of the biosimilar, because that would defeat the purpose of making it less expensive and hence cutting costs at the end of the development path. Here's an example in that same study, which was published by Smolin in the Annals of Rheumatologic Disease. That's actually the new guidelines for the treatment of rheumatoid arthritis in the European Union. You can see how that GP 2013 is compared with the reference product, and actually they do fairly similar. Again, data showing how the behavior is very similar for GP 2013 in either low-grade lymphoma or rheumatoid arthritis. And importantly, here is comparing chemotherapy combined with either the reference product or the GP 2013. And you look at the overall response rate, you look at the PRs, stable disease, and progression of disease, and they're fairly similar. I don't know, Sanjay, how do you feel? But I feel very comfortable with these data, whether I'm a rheumatoid arthritis patient or I have a low-grade follicular lymphoma. Do you have any thoughts?
No, I do. I agree with you. I do too. I feel very comfortable. You know, there's the data look very, very similar, and I mean, honestly, there really is little to suppose or predict that with any larger studies this would be any different. This is, as you and I both know, Louis, a very respectable sample size for a randomized trial that would actually be able to show a difference if there really was one.
Let's move to the next slide and see a little bit of the safety profile of these drugs. You have the combination with a biosimilar on the left column, and then on the right, you have a regular Rituximab with CVP. The incidence of adverse events is fairly similar, and as you go further down to neutropenia, constipation, and the most common side effects of Rituximab are pretty much reproduced with the biosimilar and shown to be fairly similar. The immunogenicity refers to the risk of the immune system building a reaction against the product, against the biosimilar. And the question is, are we going to induce the formation of antibodies against the product? Or is that biological going to be able to trigger antibodies against our own antibodies or our self-tissues? And we talk about the anti-drug antibodies. We see if they can be individualized to patients. And I think that Sanjay already mentioned, and I completely agree, in this setting, I find the pharmacovigilance programs to be very important and to bring a lot of comfort to us after the approval and in the post-marketing era. Sometimes antibodies can have cross-reactivity. We've seen it with several of the monoclonal antibodies that have been attempted to bring to clinic, and they develop, for some reason, unexplained thrombocytopenias and other reactions, and unfortunately those have to come off and back to the shelves. Also, immunogenicity may be associated with the loss of efficacy and induce immune effects and neutralization of endogenous proteins. I believe that those early signals in development, I think if you get a patient population for a clinical trial of 300, 500 patients, for the most part, that's going to be sufficient to see if that drug in particular is able to mount an immune reaction against it or cross reactions. The question is, what do post-marketing data tell us about biosimilars so far? I think that we are very nicely guided by the European experience. I'm also confident that the FDA is doing a very nice job communicating with the EMA and following their guidelines, and at some point differentiating and adopting paths that make sense and are safe to Americans from a regulatory standpoint. Sanjay, do you have any comments in this regard?
Lois, I agree. I think this it's very important. Firstly, this post-marketing data, it's critical, and this applies not just to biosimilars or biologics, but to every drug. And as you know very well, we learn a lot about drugs in the post-marketing era when there are plenty of patients on treatment and much more data can be collected. So it's very, very important. And I think just like you were saying, Europe is very much ahead of us in this game, and they've published this in several different formats and found no major concerns post-marketing with any of their biosimilars so far. So I'm very reassured, and we will do that in America too.
I don't know if I've told you this story before, but I was able to review the dossier that Sandoz submitted for Zarcio to the FDA. And since that was the first biosimilar to be approved here in America, I took the time to read it. And I tell you that the post-marketing data was just beautiful, was breathtaking because they had so much experience. And I think I have not done the same exercise I have to confess, with the new biosimilars. But I think that is a very nice example, and I invite the audience to, if they have a chance, visit the FDA website and go over those data. Are very nice, and it's a nice example on how to present post-marketing data to the FDA.
Good point, Louis. And I think, the good news here is that because of all the concerns that we have raised, and that's why we're talking about it, everyone is very careful, and the FDA is doing a very nice job of almost being hyper-vigilant in the post-marketing era. So I have not read what you did. Good for you that you read the whole dossier, but, it's very interesting how much information can be collected and how much more comfort we can get from that kind of information.
We are led by the European Medicines Agency, and they have about 25 biosimilars approved to date, and none has been withdrawn or suspended. And over the last 10 years, their monitoring system has been very good, and I think that also gives us the reassurance that we are on the same path. So this is another question I'd like to ask the audience to reflect and to get Dr. Agarwal's input. And the question is there a difference between using a biosimilar for supportive care or a therapeutic indication? I think that brings a good point that raised a lot of eyebrows at the beginning when we started developing biosimilars. Sanjay, any thoughts?
Louis, this is such an important issue. As oncologists, hematologists, we are very comfortable with, say, supportive care products because, after all, that's supportive care. It's important, but it's not about the efficacy in terms of a therapeutic indication. And, when I think about this and I discuss this with friends and colleagues, we know that even the reference biologic has a certain activity level, a certain response rate, a certain survival advantage, but by no means do they cure everybody, and they certainly don't work in everybody. And I think, from a patient standpoint, putting myself in a patient's shoes, I'm wondering if they're going to think that, it didn't work in me, even though the response rate may be only 30% and 70%, it's not going to work anyway, but it didn't work in me because I got the biosimilar and not the reference product. And maybe if I got the reference product, I would have responded. And that's going to be such an important issue for us to address because, patients are, after all, we are all on the same page. We want to cure them. And both physicians, that's oncologists, and the patients have to be reassured that, look, if it didn't work, it doesn't mean that the reference product would have worked any better. And, all the data information that you and I have both presented, Louis, I hope goes towards reassuring that. But this is going to be something we're going to have to proactively discuss with patients ahead of time. I mean, if I'm going to give someone a biosimilar in my clinic, I'm going to tell them a very abbreviated version of the presentation that we are giving today and reassure them that, look, remember, even the reference product works this percentage of the time, and we don't know what's going to happen to you, and therefore you must not ever think that whatever happens in the future is because there's been a change in your treatment plan.
You put it in a very, very interesting way with that last sentence. I mean, how would you explain it to patients? Because I think it's all an educational process. You and I used to have these discussions when we started doing this, and I remember my concerns and my inclination to use biosimilars more in the supportive care setting rather than in the therapeutic setting. And then the other issue was, would you care if it is first line versus second line, or if it's in the neoadjuvant setting? Those are very sensitive parts of our treatment, and where we need to make sure that all our tools are as sharp as possible. So I think my concerns in this regard have faded over time, and it's all by the reassurance of the products that are being approved and looking at the data that have been presented and the regulatory process that we have in place. So interchangeable, it's an FDA designation. It is not completely finalized, all right? But it states that a product can be substituted without intervention of the prescribing provider. And the definition goes on to mention that the interchangeable agent is that one that can be replaced with the reference product and again switch back without altering the safety or the efficacy of the product, the biosimilarity. And if that is proven, which is the challenge that most sponsors face nowadays, then the connotation is that it may be a product that will be allowed to be prescribed as interchangeable biosimilar, and state substitution laws will impact the practice. Physicians will have no say. The pharmacists will be allowed to interchange the reference and the biosimilar without physicians' discussion and approval. So with that in mind, Sanjay, would you like to discuss that a little bit more in detail?
Yeah, I think you put it very well. I must say that I think if there is going to be a concern about biosimilars, it's going to be this issue of interchangeability, which, as you very nicely outlined, is an FDA designation. I think it always scares people when you have something done, quote unquote, behind their backs without telling them. And as you pointed out, it's going to be a state-by-state discussion, designation, and legislation. We have to be aware that ultimately this may happen, and if it happens, it will be as defined by the FDA and with the understanding that the P&T committees in each hospital, and I've talked about this, Louis, how this is going to be taking place. I mean, it already happens when you write a prescription for a regular non-biological agent and you send the patient to the pharmacy, they will interchange it with something else if it's on their plan. They will do that without telling you, and it's done because there's no issue there. I think we have to get to the point where it's no issue here either, but it's going to take some time.
You're absolutely right, and it's happening, and I think that if it comes to the point where we all agree with the way ultimately the term interchangeable is sort of coined and required, then I think it makes sense that if in the health system interest it is for biosimilars to be used instead of reference products to decrease the costs of care, I think it's very reasonable. I completely agree with you.
So, Louis, again, just, a good point. And I would like to point out, as we all know, that in terms of interchangeability, we don't have to be nervous about it just yet because there's not yet a single biosimilar that has been approved by the FDA for that particular process. And we'll monitor and see if it happens, but we should be ready for it in case it does. And in my institution, what we are doing proactively, Lois, is if there is going to be something like this happening, it is informed ahead of time that, oh, by the way, if you prescribe this is what is going to be given instead, and this is why. So it's not completely blinded, and I think that's the real issue, Lois, is having the clinicians on board with the process. That's going to be very important.
I completely agree, and I think that it's going to take a very close communication between healthcare administrations and the hospitals and physicians champions in order to get this process going on. And not only that, to foster a very trusting conversation and relationship, because at the end of the day, in my mind, that matters more than anything else. When physicians feel comfortable that their administration is really doing what's in the interest of the patient. I had a patient that got prescribed Neupogen, and he was using it, and I said, he was actually complaining that it was too expensive, and I switched him to Zarzio. And it's interesting. Actually, I've had several patients. Some insurance companies did not approve Zarzio and made me go back and prescribe Neupogen. And the insurance carriers that allowed my patient to fill the prescription with Zarzio, the patients were happier because of the costs. So while I cannot tell you right now how much was the cost difference, I know it was substantially different, and it was less expensive. So that was part of my decision process. I think I'm willing to try the new Bevacizumab biosimilar, and I'll be telling you about that experience in our next meetings. How's your experience been so far, Sanjay?
So, Louis, it was a very interesting story. This just shows the disconnect, between sometimes the payers and providers and the data, that an insurance company will mandate you to prescribe a more expensive product when you can actually save money for the patient and presumably the provider and the healthcare, should I say, the insurance company itself. My experience has been similar to yours. I've been an early adopter, early embracer of this. As you know very well, I'm very convinced that the process is robust, as we've described, and I am very comfortable with biosimilars, and I am absolutely going to do what you said you would do, which is use the biosimilar products as they come to market, including Rituximab, et cetera. I see no reason not to. I'm hoping that there will be substantial cost savings. I know that one of the things that surprised me a little bit, and I think you and I have discussed this before separately, is that the cost savings are there, but it's not as much as a cost savings for prescribing a generic compound compared to, say, a chemical product. As we've shown in this program, this is a fairly complicated process to manufacture a biosimilar even get it approved. So it's not as much of cost savings as you expect. So don't expect the generic type of cost savings, but it is savings for sure. And I also feel that the more they are used and the more competition there is and the more biosimilars come out, we showed the table early on, and as we know in Europe, there are dozens and dozens more. This will drive prices down. So I don't see how this cannot be good for everybody. It's got to be good for our healthcare system, our economy, everything, that we should be able to give a patient a very similar outcome at a lower cost. So that's a very interesting story, and I hope that changes. But I can tell you that I don't think you and I differ in our approach to this. I am very willing to use a biosimilar product, and, that's a good point. Patients sometimes will drive this themselves. They will tell you that this is expensive. And I got to tell you, I am very anxiously awaiting biosimilars for some of the very expensive melanoma drugs that I prescribe all the time, as you know.
Yeah, thank you very much for your comments, Angie. With that, let's move on to a part in this presentation of patient education. And I think this is also going to be a challenge for me in clinic and a challenge for you and our colleagues. It's more administrative duties for us. So now we need to have a conversation which is not going to be simple. We have to address questions and concerns related to biosimilars. We have to educate. I think it would be great if we can have educational tools like such as videos or pamphlets or brochures so the patients best understand, and we can have our mid-level providers or the nurse navigators or ourselves answer those questions. And then we have to make informed decisions, consents, and then move on to administration of biosimilars. I think that it's a process that is already in place, but I think that is going to become more cumbersome for the reasons that you just explained, that we have to discuss with patients, we have to clear their concerns and answer every question they may have. So with that, I would love to hear your input here and your feedback, Sanjay.
I think it's very similar to what you said, Louis. This is a very important component. It is going to be some extra work for us, but I think it's worthwhile. I think it is much more important to be proactive and to discuss with the patient prior to this happening than the patient coming to you and say, hey, doc, you didn't tell me that they're going to give me a biosimilar instead of, what you wrote. And that will take much more time in backpedaling. So I think you're right. You made a very nice comment earlier about everyone, administration, physicians, everybody being on the same page, and the ultimate goal is to help the patient. And I think that's going to be so important to educate. And you mentioned communication several times. I couldn't emphasize that more. Communication is going to be absolutely key between the pharmacy, the administrator, the oncologist, hematologist, and the patient.
I think we covered beautiful and comprehensive analysis of what biosimilars are and how they've been developed over the last, I guess we have 2 or 3 years now. The biologics are very complex drugs, okay? As you opened this afternoon talking about the differences with generics, they have different structures. Their characterization is different. The degradation process is very dissimilar. The manufacturing process is different, and so are the drug patents and everything else. I think it's important to point out that biosimilars are made by living organisms, and hence differences from one lot to the other, from batches to batches, are expected and not necessarily unsafe. The comparability exercise is to be focused on a compound proving to be highly similar to a reference product. And I would ask the audience to always remember that triangle that you showed us at the beginning of the presentation, and to compare with the standalone agents where we had very small amount of preclinical data, and most of our focus were phase 1, 2, and 3 studies showing efficacy and safety. And just putting those two graphs together will allow us to best understand the process and to clear some of our concerns. And also to understand the limited number of patients that we accrue to these trials. The approved biosimilars can also have the same efficacy and safety, but remember, we are not here to prove that. We are here to show that with very, very similar analytical data, the drugs in vivo perform as well. And if we close the loop with that understanding and the objective analysis showing that, then we are in a very good position to move that drug forward. And remember that we also have to go with the totality of the evidence. That includes analytical studies, that includes PK, PDs, and the limited but very important clinical studies. And then once the FDA finds that the data we have, the data submitted, are sort of an endorsement to other indications, then we are going to have the possibility to use that agent for another patient population, something that has been termed extrapolation. Finally, we have the fact that the increasing availability of biosimilars is most likely going to broaden the access of patient populations that are underserved to biological therapies that are now very costly and that are the future, at least in cancer medicine, and I'm sure in many other disciplines of medicine, such as, dermatology, GI, med-onc, nephrology, and rheumatology for the most part. With that, Sanjay, I welcome your end comments, and thank you very much for today's presentation. Please, Sanjay, feel free to give me your perspective on everything that we talked about.
Well, I think, we've tried to communicate the importance of these agents, and I'm going to repeat what you actually said about communication one more time. This is going to be the key, making sure people are aware of what's happening, proactively going to your administration, your pharmacy, and your colleagues who know very little about this. Because I can tell you that I have brilliant colleagues, and they still think a biosimilar is a generic. And, making them understand that that's not the case, I think is going to be very important. And, it's been great, Louis, as always. A wonderful experience doing a presentation and a program with you.
Thank you, Sanjay. Likewise. And with that, I think we can complete our educational conference today.